Welcome back to another video for Bio 105. This video is going to cover the scientific method, and I am going to post this worksheet on Moodle. Your lab is at the end of the worksheet, and I will go over that. Okay, scientific method. Our learning outcomes. Outline and explain the steps. You will have to do this correctly. Differentiate between qualitative and quantitative observations and give examples of each. Formulate a correct hypothesis for a given situation. Differentiate between the variables of a scientific method. Compare and contrast independent variables and dependent variables and identify them for a given experiment. List constants for a given experiment. Differentiate between a hypothesis, theory, and law, and explain why an experiment must be repeatable. So these are your learning outcomes, or called your learning objectives for this lab. Now the scientific method differs in that many scientists include some steps, combine some steps. It depends on the science. Chemistry follows a slightly different list of steps than physiology does, or biology follows slightly different steps than meteorology. However, most scientists agree that the basic steps are the same. I have listed 7, but I've taken out 1 because we're not going to focus on writing predictions. Many students struggle with that, and for Bio 105 we don't need that. So I've brought it down to 6 steps, even though I listed the 7 here. First step is to make an observation. So let's go through that. Before I get started, you use the scientific method every day because when you go out to start your car and it doesn't start, that's an observation. When you look at the thermometer, maybe outside your window, or if you look at your phone to check the outside temperature, that's an observation. When you look out your window to notice whether it's sunny or raining, that's an observation. So you're making observations constantly. And then you follow through without even thinking about it. You use the steps of the scientific method. So let's start with an observation. All questions begin with an observation. In this step, you decide what it is you'll be studying. And I don't mean studying as in your book studying. I mean in running through the scientific method. Making careful observations is important because clear observations generate clear questions, and clear questions lead to possible answers. I'm going to use the example that my cell phone is. Our cell phone is not powering on. That's our observation. You could use the observation, it looks like it's cold outside. When you make observations, you have 2 types that you bring into play: qualitative and quantitative. Qualitative observations are made using your sense, like your hearing, your sight, your smell, your touch. Quantitative observations are made using exact measurements, like grams, meters, liters. Making the observation that our cell phone is not powering on is a qualitative observation because we're using our sense of sight. We might also be using our sense of hearing. We might press the power button and listen for the little beep, 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 beep that the phone makes when it powers on. If you look out the window and make the observation that it looks cold outside, you might stick your hand out the window and go, yep, it's cold outside. Or you might look at a thermometer that you have hanging outside and see that the temperature is -80. That would be quantitative. Sticking your hand out the window would be qualitative. Looking at our phone, listening to our phone is qualitative. If we measure the little battery, if we have a device where we can phone on the device and it will tell us battery 0%, that would be quantitative. The next step is you ask a question about the observation. So the first thing you do is to make an observation. You don't ask a question without making an observation. You can't ask a question without making an observation. All questions, and these are also called problems, are based on the initial observation and are written as, you guessed it, a question. Why won't my cell phone power on? Many people skip this step and jump right into the possible answers. However, even when you skip this step, your brain formulated a question and it gave you possible answers. So we're looking at our phone and we're going, why won't my phone power on? So we looked out the window and we said, oh, it looks cold outside. Took some qualitative and quantitative observations and we decided it's cold outside. So now we'll come up with a question about our observation. What jacket should I wear today? And your brain will go through all the different scenarios. Or you might come up with, should I drive today or take the train? Should I put my boots on or should I just wear my flip-flops? Those are questions that you asked based on your observation. The next thing we want to do is come up with an answer to our question. That's your hypothesis. It's the answer to the problem. It's an educated guess because it's written using what you already know about your problem. A good hypothesis is based on accepted knowledge and must be testable. So we can't say little green men are inside my phone. And that's a good hypothesis because that's why our phone won't power on. Never start a hypothesis with I think, yes, no, or maybe. Always want to just state it as a complete sentence. My cell phone won't power on because the battery needs to be charged. So that's the answer I've chosen to the question, why won't my cell phone power on? Your hypothesis for what to wear. So I'm going to have you do a little practice. I want you to read these different situations and I want you to circle the best hypothesis, or perhaps there's more than one hypothesis. Our question is, what factor limits how large radish plant will grow? Choose what you think is the best possible hypothesis, or maybe there's more than one, and get back to me on that. You can send me a People Moodle message with your selection. If you get it right, you might get some extra credit. I'm going to skip make a prediction, and we're going to go right into design and run our experiment. A good experiment is repeatable, and it starts with exact procedures. Now, why is a good experiment repeatable? Because in order for it to be considered a good experiment, anybody testing the hypothesis you've come up with needs to be able to follow your steps that you use to test it on their own. It's like a recipe. If you write a good recipe, someone else will be able to follow it and the meal will be delicious. However, if you write a poor recipe, let's say we're going to write a recipe for macaroni and cheese, and you say, yeah, use some pasta, use some cheese, add milk, salt, pepper, bake it, and there you go. That's not a good recipe. So a good experiment is not, yeah, you take a few things and you throw them in a test tube and then you follow through. No, that's pseudoscience. A good experiment has to be repeatable so that other scientists can test your hypothesis. The pieces of an experiment are called variables. And when you design an experiment, a really good experiment, you need to be mindful of your variables. So let's go through them. An independent variable. This is also called the manipulated variable. It's the variable the scientist, which is you, gets to change. The dependent variable is called the responding variable. That represents the variable that responds to your independent variable. So in our phone, we've said that our phone won't power on because the battery is dead. We're going to charge the cell phone's battery, and however much time we use to charge it becomes our independent variable. Our dependent variable responds to that. So if we say that we're going to charge the cell phone's battery for 10 minutes, we will then see, is the battery charged enough for the cell phone to power on? Because observations are made at the beginning and all the way through. You're going to make observations to collect your data. A control is a variable you'll be using for comparison. In our cell phone example, the control would be the same brand or model of the cell phone, but is capable of powering on. The last thing we need to talk about are constants. Constants are a group of conditions that remain the same no matter how many times the experiment is conducted. For example, if you want to know how speed affects gas mileage, then speed is the only variable that should change. It becomes your IV. The car, the tires, the road, the wind speed need to stay the same. In the cell phone example, constants would be using the same charger, using the same phone brand model, using the same charging method. Can you think of more constants for the cell phone example? Length of charging time, remember, is not a constant. It's our IV. The next step, you collect and analyze your data. Again, observations are very important. When you're collecting your data, you're going to be taking qualitative observations. So you're going to be taking quantitative observations. Is the little battery area on our phone going higher? That would be qualitative. We can measure the battery charge by using a little device we plug the phone into. That would be quantitative. You use data charts, tables, lists, any other tools to help you collect good data. Never guess at your data and never change your data to improve your results. That means that your experiment is not repeatable, and that is poor science. I'm going to actually throw this puppy on a separate page, and then you reach a conclusion. Was your hypothesis supported or negated? If you conducted a good experiment, collected useful data, and analyzed your results properly, you would know the answer to this question. If your hypothesis was supported, always use your data to support your statement. For example, you would never say, my hypothesis was supported because my data says so. Yeah, that's not going to work. My hypothesis of my cell phone wouldn't power on because the battery needed to be charged was supported, because when I charged the battery for 10 minutes, the cell phone powered on. Notice I used a number in that. You want to use your data. If your hypothesis was not supported, scientists call this negated. We never say a hypothesis is wrong, just negated. Perhaps the experiment had errors. There could be a lot of reasons why a hypothesis was negated. If your hypothesis was supported, that doesn't mean that your hypothesis is 100% beyond a shadow of a doubt the explanation. However, you may say the hypothesis is supported. There are no absolutes in science. Although a hypothesis that is constantly being tested with new experiments and continues to be supported will become theory. And theories that stand the test of experiments eventually become laws. But remember, there are no absolutes in science. Even laws can be negated. So can you think of any theories in science? Can you think of any laws? Can you think of any theories or laws that new scientific data have negated? Think about it. Okay, Lab 1. You're going to type your responses to the following situations, and you're going to submit your answers as a PDF. You're going to include your name, section number, and your answers will be in complete sentences. If your responses are correct, but you did not write in complete sentences, you'll only receive half credit. Then you're going to take your thinking further. Using your own words, explain why it is important for an experiment to be repeatable. Your response must be written as a complete paragraph. Do you know what a complete paragraph is? A correct response not written as a complete paragraph will only receive half credit. And that's it. Ciao.